Music.
Okay. All right. Good morning, everybody. I hope everyone had a nice weekend and we're working our way through the plant kingdom. Let's see. All right. Let me set this up. There we go. Cool. We'll just jump right into it. If anyone has any questions about anything, just feel free to throw it in the chat box. Could be about anything, but I'll get started, but just because I want to give you guys opportunities to like ask me questions about logistical things, or anything we've covered so far. So don't hesitate to ask questions in the chat box if you think of something. We're gonna first finish. Let's make this big. Let me fix this too. There we go. Move that up. Okay, first we're gonna finish up. This is where we left off last time. Actually, we left off here last time, talking about how that shift from homospore to heterospore was like a huge evolutionary shift in the plant world, right? A significant shift, and a shift that ultimately ends up with the seed shedding plants, which we'll talk about later in this class. Not this class, but later in this course. Before we move on to the other spore shedding plants, well, first I want to talk just a little bit about this fossil tree. This fossil tree called Lepidodendron, which even sounds fossil-y, in my opinion. It sounds like a dinosaur tree. So you might have remember in the beginning of last class, we kind of talked a little bit about the Carboniferous period and how significant that period was for the formation of coal and other fossil fuels that we use now. But if you were to go back in time, right, over 300 million years ago, and you were in the Carboniferous period and you were in one of these forests, you would probably see a lot of these trees, which were actually lycopod trees. So remember the last class we talked about the phylum Lepidophyta, which included the genera Lycopodium and Selaginella. Well, this was an ancestor of those, and it's called Lepidodendron. And this here is what the sporophytes of that, so this is all based on the fossil record. Here's what the sporophytes of that extinct tree would have looked like. I'll annotate because, here, let's change colors. So when the sporophyte first emerged, it kind of almost looked like the Lycopodium or one of the other, yeah, mostly like Lycopodium, like some of the other equal dichotomy type things we looked at before in this last class. But then, remember, I said that during the Carboniferous period, there was all these tropical conditions for a lot of the land mass. And so plants could grow for a really, really, really long time, long growing season, because it's so tropical. And so these little guys here, over time, would actually get to be kind of like more of a tree-like shape. It almost looks like a bottle brush or something, until ultimately grows into this mature tree. But you can see even here with this mature tree, the top of it kind of still has that equal, so we have equal dichotomy at the top and then unequal dichotomy otherwise, because it's, Mohammed, you have a question, hun? Nope. Okay.
You just unmuted.
Okay. Alrighty. Yes. So the point is that it's, even though it became a giant tree that was about 100 feet tall, give or take, and about 6 feet wide, and that's pretty giant tree. It still kind of had a lot of the characteristics of the other Lycopodiophyta members that we've looked at. including the fact that there's equal dichotomy at the top of the tree, and at the top of the tree there's also strobili. The book says cones, that's not right, but strobili, which is what we saw in the other members. Also, a similarity is that they had microphyls, right? A vein with a leaf with a single vein, small. These grew right on the stems. So this here on the left is the trunk of this tree, and all of these things coming out of it are the microphyls. And just like a lot of trees in our region, including which we could really see this time of year, the leaves would fall off every year. So the tree was deciduous. And when they fell off, they left these kind of diamond-shaped marks on the trunk of the tree. And this is a fossil showing you that. So let's go back real quick. So this here, you see here it says vascular bundle scar. It's this little dot in the middle. Here's another one. That's where the leaf would have attached, but it leaves, like I said, this kind of like diamond-shaped pattern on the trunk of the tree. And in the fossil, we actually see exactly that. So here's a fossil of a tree trunk from Lepidobidendren. And here I'll change colors just in case that doesn't come up very well. Oh, that didn't really come up as well either. But anyways, you see all the little diamond shapes. Okay. Here's one of those fun reconstructions, right? So you have like the young trees that look a lot like the members of the genus Lycopodium, kind of that brushy, fuzzy, stalk-like thing. There's a mature tree in the background. And then hanging off the tips of the, oh, here's a trunk. And then hanging off the tips of the top of the tree, you'd have these many strobili, or here's one strobilus. And we know from the fossil record that Lepidodendron plants were heterosporous, so they had microspores and megaspores. And one cool thing about this fossil tree is that the megasporangia, which are located on the megasporophylls, remember, like mega means female in the plant world. So megasporangia is the sporangia that's going to release the megaspores, and they're located on megasporophylls. But the megasporophylls actually served as like little boats in this carboniferous swamp. So here is a megasporophyll, and on top of that, we'll do it in blue, I guess. See this megasporangium. And so that actually would float on the surface of the swamp water. And then whenever the microspores were released, they would fall directly on top of the megasporangium. Sorry, of the megaspore, once the megasporangium opened. Yeah, well, it's not very exciting, I have to admit, but it's kind of cool. It's a little boat. Okay, that is it. That's the only thing we didn't cover last time for lycopods. Now we will go into the horsetails, and I think some of this we may gloss over today. Some of this I might not care about you guys knowing too much about. We will talk a little bit about the stem anatomy of the horsetails, and so we will have to talk a little bit about cells and tissues and Sorry, something popped up in the chat box, but I don't see it there anymore. No, it says here, hold on one second. Let me just see if I can pull up the chat box.
I was just asking if this is
lecture 7. So so just keep in mind that the here, do I have it handy? No, I don't. So 6 and 7 are combined. So we just finished 6/7. See here, it's because, so this is technically 6 and 7 because it covers the Lycopodium and then it covers Selaginella. Do you see this lecture slide here, the lycopods, diversity, morphology,
Yes.
all that stuff? Okay, cool. Yeah, so in sometimes in the past, I've done this in 2 separate lectures, but I smushed them together, and so now it's just a single lecture. So for example, the next assignment that is due on Thursday covers the introduction to the vascular plants, and then, so it says lectures 5 through 7. It only covers this lecture, the lycopods, and the previous lecture, which is on the introduction to the vascular plants. If you pull up the slides on Blackboard, it'll actually say, it'll call this lecture 6 and 7 on Blackboard.
Thank you, Professor.
No problem. Okay, let's move on to 8, lecture 8, which is the horsetails and the scouring rushes. Yes, Equiseta. We'll talk about it. Yeah, we did that last time and finished it up today. Today we'll talk, like I said, a little bit about the steel, which, you know, anytime we talk about stem anatomy, I'm like, yay, but, and then we'll talk about alternation of generations in the horsetails. And then I don't think we'll cover the evolution of megaphily today, but we might. I'll keep an eye on the time. Did I bring my, I did not. It's okay. All right, here, actually, let me just make that, yeah, 10, 12. All right, so we're doing okay on time. All right, so this is what horsetails look like. So this is in the phylum Equiseta, and they look like horsetails. Another thing people call them is the scouring rushes. So this is how you pronounce that phylum name, Equiseta. The reason we, oh, and there's a lot of diversity, but they all kind of look very similar in structure in that they have the spore-bearing things on the top of the stem, but sometimes you get a lot of branching patterns. So for example, you know, this one here makes a lot of branching, whereas the one underneath here, not so much, right? So that's really the main difference of some of these, but oops, let me make this small. Okay. They are called articulates because they're jointed. So if you live in New Jersey, you actually come across these quite a bit, even around Newark. I've seen these a lot. And near the railroad tracks a lot of times is if you ever take New Jersey Transit. And like the stems kind of like break apart, almost like Legos or something. They snap off at these areas here. Here's an area, and you can see here where one is snapped clean off. And because of that, we call them articulates. Why Equiseta? Well, people call them horsetails because the name Equiseta, so equus means horse, and then seta means hair. So whoever first described this plant phylum thought that they resembled a horse's tail. And you can see here, even in the animal world, if we were to look at the genus Equus, all of these different animals are in that genus. So zebras, donkeys, and of course horses. So, you know, people didn't have as much going on back then. So I guess horsetails, fine, fine. Here you go. Kind of looks like a horsetail. It really does depend though if it's more of a fluffy branching one versus a stick-like one. Very similar to the lycopods, right? The phylum Lycopodiophita. Equiseta also arose around 400 million years ago and really exploded during the Carboniferous period. So they were another large contributing member to the formation of coal and fossil fuels, and dominated those swamp forests of the Carboniferous period. This on the right is showing you, on the left, you're seeing a reconstruction of a horsetail forest, and on the right, you're seeing an actual fossilized specimen of a horsetail from that time period. Of course, we're in the vascular plants, so we know the sporophyte generation is the dominant generation. And in the phylum Equiseta, there is only one genus, which is convenient for us, makes it easy to remember, and that's just Equisetum. There's about 20 species in there, and as I mentioned, there's several that occur naturally in New Jersey, including Equisetum hyemale, which is what these images show you here. There's a real picture on the left and then a botanical drawing on the right of Equisetum hyemale. And then there's Equisetum arvense, which both of these occur naturally in New Jersey. And this is what I was saying before. Like in terms of horsetail, Equisetum hyemale doesn't really look like a horse's tail. Like if your horse's tail looks like this, your horse is probably sick. It shouldn't look like a stick, right? It shouldn't look like a stick. But if your horse's tail looks like this, okay. So probably whoever first identified or described these plants probably saw this fluffier version, I'm hoping, or they had some pretty sick horses. We don't really use horsetails for anything. We don't make money off them. Sometimes actually they become problematic in that if animals eat them, they can sometimes unfortunately die because the actual plant contains silica. It's not good to eat silica. So sometimes people make efforts to remove them if it's growing near livestock. And you'll find them in a lot of different habitats. They're not too picky, but usually in areas that are kind of sandy or growing in water, but not submerged or floating. It's something we call emergent, kind of like a cattail. Maybe you've seen a cattail before. Another thing, horsetail, cattail, never thought about that. Where it's growing in the water, but it like sticks out of the water. That's called emergent. But particularly you'll find these in disturbed areas, right? Like I said, I see them whenever I ride New Jersey Transit. I would always see them growing along the sides of the tracks when the train was stalled and we're waiting for something, right? Because that happens a lot on New Jersey Transit. I don't miss that. I do not miss my commute from Brooklyn to Newark on a regular basis every week. I don't miss that. There's a long commute. All right, what else do we know about this? All right, yes. So we're in the vascular plants. So we know that the sporophyte generation is dominant. We know that it has vasculature, right? Because we're in the vascular plants. We know that it has true roots, stems, and leaves. And for Equisetum, right, the only genus in the Equiseta phylum, there is only one type of sporangium. So we're back to talking about plants that are homosporous, right? Homosporous and sporophytes, and the gametophyte will grow outside of the spore. We learned this term last time. Exo means outside of. Spore refers to the spore. So sporophyte plants are homosporous. Gametophyte plants are exosporic in the horsetails. Okay, and then growth. Okay, yeah. So the reason I'm showing you this, I think it comes up in lab. That's the main reason I'm showing you this, is that the growth that's occurring is happening at the tip of the stems, kind of like we're used to. And the main meristem that is driving the growth, oh, you know what, I bet you I give you it here. I don't have to draw it. Is this like upside down pyramid looking thing? Here, let's see if I do draw it for you guys. I probably do. Okay, so I'll give you a diagram here. So this is a microscope slide. This is a diagram put on top of that. And you see that kind of polka dotty part? That is the meristematic cell. That's the meristem. And that cell is going, remember we talked a little bit about meristems before, in that they're kind of like a stem cell. So like a meristem can grow can divide and grow into any different type of cell, which ultimately will make any different type of tissue. So you can think of a stem cell when you think of a meristem. And so it's at the tip. And as it divides, it's going to grow things like the stem tissue. And you see here a leaf tissue. So as it grows and divides, that's what it's going to be leaving behind it. Other parts of the stem. But just so you know, I ended up not drawing it. I thought I did. It's here. See that upside down pyramid? And that's that meristem cell. Okay. What else do we care about? It has a rhizome. Remember, rhizome is just a fancy word for horizontal stem. So the stem here, look, stem is growing horizontally. And then every now and then you'll get vertical branches coming out, right? Like you see here, right? Vertical branches coming out. Okay, let's see. Dimorphic. Okay, we might as well mention this word now because it will show up in future classes. So it says here, some species have dimorphic upright branches. That just means two types or two forms. So the di means two and morph refers to a form. So dimorphic means two forms. Some species of Equisetum will have two types of branches. One branch will be green and not produce any spores. So we say a sterile green upright branches. And their main job is going to be doing photosynthesis. Then the other branch will look kind of like this, and its main job is to have, that's actually, trying to think if that's, don't worry about, don't look at that word right now. Just know so sorry, I'm getting distracted. One branch is green, usually green, and does photosynthesis. The other branch typically is not green, but its job is to release spores. And don't look at that word because this is a new thing now. I gotta update that. Okay. As I mentioned, yes, so they're photosynthetic stems. The stems themselves are green. Notice something here, right? Where are the leaves? Where are the leaves on these plants? They're not very obvious, and even more so, the stems are green, right? So the stems are going to be the main thing that's doing photosynthesis in the horsetail. And I talked before about how you can pull the stems apart, and that's why we call them articulates. When you pull them apart, you're pulling them apart at these areas called the nodes, which I think I draw here, but like here is a node, here is a node. Inter means between node. So the area between the nodes, that is right here, that's the internode. I think I draw it on the next slide. Yep, or this slide there, node to node, and then internode is the space between the nodes, right? So we're still talking about the stem here. Contains silica. So if you were to touch a horsetail stem, it almost like feels like you're touching like this pencil. It's like this is a plastic pencil. It's very rigid. It's very hard. It almost feels like it's man-made. It almost feels like it's man-made. It's very almost plasticky feeling. And that's because the plants contain this silica in them. And that serves, the reason they're like that is, remember I said they grow in water, but they stick out of the water. So like if there's currents in the water and kind of like moving them around, being so rigid and stiff kind of helps protect them against getting destroyed by running water that they might be growing in. What humans have in the past used this for, and this is another reason we call it scouring rush, is because you might, have you ever, maybe, I've gotten better with cooking during pandemic, but you know, have you ever done this, you know, like where you're trying to cook a meal and then maybe you get distracted and whoopsie, and down the bottom of the pan is a hot mess. So nowadays, when something like that happens, most likely something like this, right? Like was that steel Brillo pad or something like that. But in the past, in the very past, before we had the conveniences of things like steel wool, people actually use scouring rush to scour pans, to remove this baked on mess when you make a mistake in the kitchen. And so that's why people also call it, in addition to calling them horsetails, people also call it scouring rush. And for the record, I mean, if you're ever camping or something and you burn your pot, you can still use it for this purpose. It works very well. The reason it works so well is because, again, it contains this silica. Do I care about this? Not really. It's just letting you know that there's extinct members that look a lot like the extant or living members of steel. All right, I'm going to talk about the steel, but I'm not going to go too crazy on this because we do talk about it in a lot of different classes. But horsetail has a different kind of stem arrangement in terms of cell types, so we should at least talk about it a little bit. So remember, a stele is a word that just talks about how the xylem and the phloem are organized inside of a stem. That's all it means. Oh, chat box. Oops. Why does my chat box? Here, let's see. Here we go. Scouring rush is a genus in horsetail, or is horsetail the genus of Equisitophyte, and scouring rush and horsetail the same thing? So here, I'll write it in the chat box. So Equisitophyte is the phylum. And in the phylum, let me close this. In the phylum Equisitophyta, there is only one genus that remains, and that genus is Equisitum, which is the genus, the only genus that remains in that phylum. In Equisitum genus, there's about 20 species, and we commonly call those 20 species horsetails or scouring rushes. So horsetails is a and scouring rushes equals the common name of species in Equisitum. The horsetails and scouring rushes are the same thing. They're just a common name. Some people prefer to call them horsetails. Some people prefer to call them scouring rushes. I think most people call them horsetails. Yeah. Yeah. Anytime you have questions, I'm really happy to answer them, because I don't want some of the stuff can get a little, there's a lot of words and they're kind of all flying around and can get a little complicated. So it's good sometimes to like take a step back and be like, all right, what are we talking about? She's talking about horses. I don't know. So good to kind of regroup. Yeah, so the steel. So there's 4 types of steel. There's 4 steel types. I don't know. Steels? I don't know if that's right. There's four types of steel organization, right? There's protosteel, siphonosteel with leaf gaps. Sorry, siphonosteel with no leaf gaps, siphonosteel with leaf gaps, and then U-steel. We will talk about those other ones as we move along. And we did mention for example, in Lycopodium, when we were talking about the lycopods, we did mention that those earliest vascular plants, like the Lycopodium, have what's called a protosteel. If you were to draw these, sometimes this helps. I'm sorry, I have like frog in my throat. If you were to draw these, this is how you could think of these. So here, a protosteel is like a solid thing, and inside the solid thing is xylem and phloem. Here, I'll do this. We'll do blue. So like inside this is, you have xylem in the middle, sometimes in this kind of star shape. And then around that, oh, come on, annotate, not working with me today, is the phloem, right? But the point is that it's solid. So when I say solid, I mean there's no hole or anything. So think of this whole thing, whoa, that didn't work. Think of this whole thing as being completely solid, right? Solid, right? That's a protosteel. And things like Lycopodium had that. Siphonosteel with no leaf gaps. Siphonosteel just means that there's a hole in the middle. So you can see here, the xylem and phloem will be on the outside, kind of like in this ring shape. But in the center, there's a big hole. So that's what siphonosteel kind of looks like, and no leaf gaps. Later on, we'll look at plants. It'll still kind of be like similar in look, but then you'll see other things around it, like this. And those are the leaf gaps. And then a eustele is like little. So when I'm drawing these red things show where xylem and phloem is located, right? So like eustele kind of is like rods. Boy, that is one ugly drawing, but either way. So yes, Lycopodium had protosteels. Lycopodium had that simple, solid version, right? And this is what we saw when we did that class. Later, there's like a little slightly more evolved in Selaginella, in that they have the second one, which is the siphonosteel with no leaf gaps. Now we're in the horsetails, and the horsetails have kind of a combo. It's like a mix of this one and this one is what the horsetails have. It's kind of like a mix. So we say it has a eustele like, I know this is a mouthful, eustele like siphonosteel. Real mouthful. But here, I'll just show you what I care about this. How about that? I'll just show you what I care about that. So this is a cross section through a horsetail stem. There's some things here that we've already come across, right? So the outside has an epidermis. The outside also has areas of gas exchange, which we've come across before, the stomatal apparatus. And then I mentioned the silica a bunch of times already. The outside cuticle on that epidermis secretes the silica. Okay, that's the epidermis. Oh, those red circles are showing you where the stomatal apparatuses are. Okay, I don't care about this right now. This is something you guys can cover a bit more in lab. But the point is there's a cortex, and the cortex has a bunch of different cell types that I don't care too much about today. But I do care about these canals. So this is a close-up of that cortex. So just so you know, don't get disoriented. Look, so here's that cortex is right here. And on this next slide, we're just going to look at it closer. Right, closer. So, all right, I guess I do care about this. Fine. On the top of the cortex, you see this darker stuff. That's the fiber cells, or you can use the mouthful sclerenchyma, but these are the fiber cells. And they help keep the plant upright. So they're for mechanical support. You can think of them as like a scaffold. The green stuff is the parenchyma tissue. That's where photosynthesis is going to occur. I'll go over these terms right now, but they'll come up again in lab when you get to this lab. So I won't spend a ton of time on them now. But this is important, is these follicular canals. Follicular canals. So even if you look here, right, you see these kind of holes. There's a lot of holes. There's a lot of holes. And many of those holes are these canals. So if you were to, I know maybe it's not super clear, but like what I'm trying to point out in this slide is these big giant holes. These big giant holes. And this on the right is showing you close up. And what those holes are they're basically just almost like an empty straw in the plant. And remember, the plants are submerged in water, and so this helps air get from the top of the plant down into the bottom of the plant because it needs air for stuff to occur. I don't care about endodermis for now. All right, you still like Siphonosteal, right? You still like Siphonosteal. So what do we know, even just from my brief little thing in the beginning? A Siphonosteal is like, has a hole in the middle, right? Remember, if I'm doing this red, the red is like where xylem and phloem are located, right? But oh boy, oh boy. Siphonosteal means there's a hole in the middle, right? Right, so you see in the middle, there's a hole. That's this part, Siphonosteal. Eusteal is like rods, so if you were to look at the top of the rods, they would look like this. They would look like this. And each of these little rods contains xylem and phloem. That's the eusteal part. So look here, I say it right here. Vascular tissue arranged in vertical rods, and the rods are in a ring. So let's just see if I draw it on the next slide. If not, I'll draw it here, but hopefully I did draw it. Nope. So just so you see, Here, all right, so arranged in vertical rods, vascular tissue arranged in vertical rods. Here's one, here's one, here's one, etc. Going all the way around, right? Going all the way around. Here, look, here's another one here. Here's another one here. That's the arranged in vertical rods. But notice the rods are also in a ring, right? So these rods are in a ring, and there's a big empty hole in the middle, right? That's the Siphonosteal part. Rods are the eusteal part. The ring is the Siphonosteal part. So this is cross section of a horsetail. This is just to show you here, right? We're talking here about the vascular tissue. Here is, remember I was saying there are rods that are in a ring. Here's one of those rods. If we were to look closely at that here, this is one of those rods right there. These are what it has in it, right? It has xylem and phloem, which are labeled, but just in case you can't see, the xylem is right here. The phloem is right here. And then there are these littler holes, which are called the carinal canals. We don't really know what they do, but they help us find the xylem and phloem. So like here's remember these bigger holes right here, whoa, and right here are those follicular canals. And then there's these littler holes, which we don't really know what they do, which are carinal canals, carinal canal. But the nice thing about the carinal canals is they help you find the xylem and the phloem, because wherever there's a carinal canal, there is a xylem and a phloem right by it. So for example, on this previous slide, just to get you oriented, We'll use a different color, green. This big thing, that big thing, those are the vallecular canals that help with gas exchange. And these small things here, those little holes, are the carinal canals, and we know the xylem and the phloem are located right by those. The reason why the vallecular canals are called vallecular canals is because, do you see how the outside of the stem kind of goes in a hill and then a valley and then a hill and then a valley and then a hill and a valley, etc., all the way around? The vallecular canals are located in the valleys. See here? Valley. That's why they call them vallecular canals. Valley. Oh, can you tell how much I love talking about stem anatomy? Whee! Okay, so you still like Photosynthetics, right? Means vascular bundles are in a rods and the rods are in a ring. And that ring is the siphonosteel part. If you're a ring, it means you have an empty part in the middle, right? If you're a ring, you have an empty part in the middle. The empty part in the middle is called the pith region. So the pith region is all of this part here. That's the pith region. All right. Yay, I love talking about stem anatomy. All right, here we go. This is just showing you from the fossil record. This is that one I showed you at the beginning. It's an extinct member of the horsetails. But the point is, look, here's a fossil from that extinct member. And what do you know? It has all the same things that we just talked about. Same exact anatomy, same canals, has a pith region, has valleys, etc. So that's just showing you that for over 300 million years, the horsetails have been relatively unchanged, except that nowadays they're very small, whereas back in time they were much bigger trees, but otherwise the same. Right. Do I care about this? All right, well, remember I said that some of them look like a horse's tail and that they're fluffy, kind of branched, and some of them don't. They kind of look like sticks. Well, if they are branched, the branches will come out at the node. Don't worry about primordia right now, but just worry about nodes. Nodes are those regions where you can pull it apart. That's where these branches are going to come out. So the picture on the right shows the branches coming out. Question in the chat box. My chat box is just not behaving today. Let me pull it up here. Here we go. Are the branches photosynthetic? They are. They are photosynthetic. Yep. So the stem and the branches are photosynthetic. But again, you might be thinking like, well, where are the leaves? I thought this is a vascular plant, right? We said it has true roots, true stems, true leaves. Well, I don't see any, I haven't seen one leaf yet. Yeah, don't worry about that. Leaves. Well, because they're, believe it or not, we've been looking at them the whole time, but they don't look like a leaf at all. So you see these little things here at the nodes, these little, here's one. Here is, I can't even draw it so small. Here's one. Here's one. Down here's another type. Here's one. Here's one. Those are actually the leaves, believe it or not. And they are very small. They're simple. They have one vein. And if branches were going to show up, the branches would come out right underneath the leaves here at the node, if the branches were going to be there. And if you're looking at these leaves, you can notice right away they're not green. So the leaves in the horsetails do not do photosynthesis. They're not photosynthetic, the leaves. They're not. Let's see. Yep, leaves. Okay, I just want to see, what else do I have with this today? Yeah, we're almost done. I don't want to spend too much time on, right. Okay, well, we're gonna talk about this a lot next time. I think, yeah, we'll talk about megaphily next class, but one thing just keep in mind here, this is a little confusing, but you see how it says small and simple and one vein. If you've been paying attention in this class, if you saw these terms together, you would think, oh, it's a microphil, right? Because that's what we've already talked about many times, is this idea of a microphil. Well, even though these share the similar traits of being small and one vein and simple, these are actually not considered microphils because they probably were originally a megaphil that we'll talk about in another class. I'm trying to think of how I can, okay, here's how I can explain this. Here's how I can explain this. What we think happened in the fossil record, what we think happened is that at one point, this probably was like a leaf that looked like this. And this leaf that looked like this probably had a lot of veins, a lot of different veins, and was not a microphil, right? Was not a microphil. But over evolutionary time, a lot of this leaf was kind of lost, such that now we just have these things left, and we call these things leaves, but they probably have an ancestral basis in being a larger leaf that had many veins. This might not make sense as like, why would we care about that? The reason is because megaphils, which you could see this word here, megaphils and microphils evolved from different ways. So that's why we make that distinction. We'll talk about megaphils next class. I don't want to talk about megaphils right now. I want to get to the alternation of generations. They have roots, and just like we saw in Lycopodium, the roots come from the stem. The roots come from the stem. And remember, rhizome just means horizontal stem. Okay, finally. Okay, I just wanted to get to the alternation of generations part because that's basically the end. Here we go. So this is what the gametophyte looks like. Now remember at the beginning of the class, we said that the sporophyte is homosporous, so it only has one type of spore. And we know that if a sporophyte sheds one type of spore, then the gametophyte that grows out of that spore is going to literally grow out of that spore and be exosporic, right? And that means your gametophyte's going to be your little green blob that we're used to seeing. So here's a little green blob of a horsetail gametophyte. Remember, we said if the gametophyte grows outside of the spore, that it's going to have both sex organs on it. And we can see that here, the antheridium and the archegonium on a single gametophyte. So very similar to what we're used to already, right? Simple thallus, photosynthetic, small, both sex organs. Yeah, usually. The reason why bisexual usually is because sometimes it will stagger which sex organ is on it. So you might come across them and it has one sex organ or the other. And you would think, oh, well, this is unisexual, but the other sex organ will show up later. So it's usually bisexual. Just for your purposes, think of it as bisexual. Oh, right, that's what I just said. Right. Think of them as functionally bisexual. Think of them as functionally, think of them as bisexual, but sometimes the sex organs develop at different times. Sometimes the sex organs develop at different times. All right, let's just go through, this is all I want to get to. Let's go through this alternation of generations in the horsetails. Okay, because there's a lot of things here that we're already used to seeing, and so it'll feel a little bit more familiar, right? All right, so egg and sperm already came together. and we have our 2N zygote. That's going to grow, 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 right? Remember, green here is diploid, right? Green here is diploid. So the diploid sporophyte generation is going to grow, grow, grow until we end up with the mature sporophyte plant. Here's the things we've already come across, right? The rhizome, that's that horizontal stem. In the case of this representative species, we have the two types of branches, right? Dimorphic branches. This branch over here just does photosynthesis. This one over here, its job is to produce spores. Oh yeah, okay. So we say if its job is to produce spores, we can say that this is the fertile shoot. Look, here's a node. Here's another node. Here is another node. Here's the space between the nodes, right? Or the internode. And then at each node, sometimes you have branches like you do here, or you have leaves, which you have here. Okay, so that's the basic parts of a sporophyte plant. This is where I was getting confused before. Okay, so remember back on the other slides where I like crossed out strobilus? It's okay, we can keep strobilus. Strobilus is okay. Because I was getting confused with this new term that we're going to talk about. So at the tip of the plant, the tip of the sporophyte plant, you have a strobilus, which we're used to. If you were going to do a cross section through or longitudinal section through that strobilus, we have a new structure to deal with, which is Sporangiophore. Remember, phore, we've come across that term before. Phore means to bear or to carry, right? To have. And so this is a structure that bears or carries sporangia. And so each of these, they kind of look like mushrooms. They kind of look like mushrooms. So each of these things, so here's one. Here's one. Look, on this side, here's one. Each of these things is what's called a sporangiophore. Here's one that's been pulled off. Sporangiophore that's been pulled off. Sporangiophores make it so that the strobilus can have way more sporangia because each sporangiophore has several sporangia on it. So here's the top of the sporangiophore. Here's the stalk of the sporangiophore. And each of these, I'll use a different color here. Each of these things hanging off is a sporangium. So one sporangiophore can have many, many, many sporangia. And so this makes it so that in the horsetails, one strobilus can bear a lot more spores than we've seen previously in the plants. Okay, so in the sporangia, that's where meiosis is going to occur, and your result of meiosis will be your haploid spores, right? So there's your sporangiophore. Here this was a drawing. Here is it in real. Here it is in real. This is one strobilus. Each of these little mushroom looking things, right? There's one, there's one, there's one, there's one. Each of those little mushroom looking things is a sporangiophore. Here's a close-up of two sporangiophores. And on each one, there's many sporangia on each one. So it just makes spore shedding in the horsetails way more efficient because you have so many more spores that you can release than if you used simple sporophylls. Now we have sporangiophore. It's like a whole structure. So there's your sporangiophore. Your spore is going to get released. The spores have something we've come across before, which is like these little kind of filament things that help the spore get out. Spores are going to land on the ground and germinate. These are close-ups of the spore. And each of these kind of filament looking things are those elaters. They just kind of, they're sensitive to humidity. So when the humidity changes, they kind of like move around. It helps the spores get out of the sporangia. All right. Spores are going to land. They're going to germinate. They're going to grow into the gametophyte plant. Gametophyte plant has on it both male and female sex organs. Male sex organ is going to make sperm. Female sex organ is going to make egg. Eventually, the sperm is going to leave. Here's an antheridium. Eventually, the sperm is going to be leaving the antheridium, swim, 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 and fertilize an egg. This is a close-up of a sperm right here. Once egg and sperm fuse in fertilization, then you are back to your 2N zygote. So not that many different players than we've already seen, right? Not that many different players. We've come across strobilus before. We've come across leaves and roots and rhizome before. We've come across sporangio before. The only thing that is new here now in terms of the horsetails is some of the stem anatomy is new, but then the sporangiophore, right? Sporangiophore is the newest addition that we learned of today for this guy. Some people use horsetails for different herbal medicines. So like it has traditionally been used to do all of these things here, right? Heal wounds, maybe treat kidney problems and tuberculosis. But a lot of the modern studies that have been done have not been in humans, they've been in other animals. So just so you know, if you're interested in learning more about that, there's a paper there that you could read about that. But overall, if I just go back here real quick, overall, when we talk about the horsetails, there's not that many different things to remember. It's very similar, except for parts of the stem anatomy and this new structure that we call sporangiophore, right? That's the main new thing. Yeah, you have things like node and internode, but node and internode will happen again in other classes. We had like two types of stems, right? A vegetative stem and a fertile stem. Yeah, and the stem anatomy, but overall, a lot of the same players that we've seen already. Okay, horsetails, not my favoriteest one, in case you couldn't tell. But next class, we'll start talking about the ferns. Ferns are my favorite, probably. I think my favorite. And there's a couple classes on ferns. So Thursday, we'll do ferns. Just a little nice reminder that you guys have an assignment due by Thursday before 10 a.m. Remember, this assignment was originally due today, but I pushed it back so you guys had more time with the material. So what I would recommend that you do for these assignments is to study the material beforehand, make notes, think about some of those important terms, go through the slides, and then attempt the assignment. You should definitely be studying before you do these assignments because they're timed. This one will have more time than the previous have. I think you'll have a half hour to complete it, a little bit more time. But just keep in mind in the syllabus that if your assignment is late, it's automatically dropped a letter grade, and that if it's more than 2 days past the due date, I don't accept it. So please turn your assignments in on time. They're to help you learn the material. Got a question in the chat box and I can't see the chat box. Are all the assignments being pushed back now? No. Here, let me stop share. No, they're not, because here, let's pull this up. Where is this? I'm trying to pull up the syllabus real quick. Here we go. And let's make this big. And here I will share the syllabus with you guys real quick. This is the most recent version of the syllabus. It's on Blackboard. Make this a little smaller. Can you guys see? You guys can see the syllabus, I'm assuming. Let's see where we're at here. So, all right. So we just finished. Today is the 16th, right? That's this. And we just finished up the horsetails. Horsetails we're done with. Next class, which is Thursday, you have assignment 3 due. And assignment 3 covers lectures 5 through 7. So that's Thursday. We finished that material last Thursday. So the assignment that's due on this Thursday covers these courses. The only thing is this lecture kind of, I think it went into the next class. Like I think we covered lecture 5 a little bit on this day and a little bit on this day, such that we covered lycopods all on the 11th, I think, or a combination of those two. We over the course of 3 classes. Here, this isn't, but yeah, this assignment that's due on Thursday covers introduction to the vascular plants and the lycopods. But these here are in a single lecture. This is one video, one recording, one slideshow. So it seems like it's a lot, but it's not. It looks like it's more than it is when you see 5 through 7, but it's only 2 classes, I guess is what I'm trying to say. So that's due Thursday. Look, there's no assignment after that until after the first exam. So this is a good reminder. The first exam is the 2nd of March, which is 2 weeks from today. 2 weeks from today is when the first exam occurs, right? But that exam only covers through the horsetails. So exam number 1 covers this stuff that we finished today. Exam number 1 does not cover ferns, which we will talk about next time. I don't know if that helped or made things way more confusing. I see something in the chat box here. Let me stop this. Chat box. Oh, sorry. Happy birthday. Ooh. Sorry, Catherine. Yeah, any other questions? So 2 weeks from now, please, how many questions? How many questions on what thing? How many questions on this upcoming assignment? Around the same that there's always been. So probably exam is not written yet. So I don't, it's occurring 2 weeks from now. And I think that's very nice that you think I have it, my stuff together so well that I have something that's due 2 weeks from now already completed. But the exam is not written yet. It's 2 weeks from now. So I don't know how many questions are going to be on that. On the assignment's been up since last Thursday, I think. Something like that. So yeah, it's around the normal number, right? Like 15-ish. Are the exams online? Yes, the exams are online, and the exams are going to have very similar format to what the assignments have been like. That's another reason I do these assignments, is so that you guys get used to the types of questions that I ask, the formats of the questions. Doing the assignments on Blackboard, the exam will be that same type of format. Oh, this is a great question. Is it at the same time of the lecture? That's a great question. So the exams are going to be similar to how the assignments are, in that the exam will be available for a block of time. So for that class, here, I'll pull this back up. I will pull this back up, syllabus, and share. Right, so for that class, that's 2 weeks from today, right? 2 weeks from today. We won't meet for lecture because you'll have an exam. And what will happen is the exam will be available starting around the time of lecture, but it will be available for a block of time. I haven't decided what the block of time will be yet, but it'll be similar to the assignments in that it's not like a certain exact time. It's that it's available and it's due by a time, right? Okay. I hope that cleared that up. All right, any other questions? So assignment 3 is due on Thursday. Turn it in. It's the last time you'll get a chance to see feedback before the exam. And we'll have a little bit of a review, I think, before the exam as well. Is assignment 2 responses open yet? Did that open? Were you able to see your answers or questions for assignment 2? Did anyone try to look? No. All right. Well, I'll check to see if assignment 2 should be open and available already for you to see what you got wrong. If not, I will go check right now and see that it is. Okay. No, we can't. Okay, cool. I'll fix that now. I thought that it automatically would allow you to, but I guess it didn't. So I will fix that right now. Okay. All right. If anyone else doesn't have any questions, I will see you guys on Thursday. Turn in your assignment, please. It's available now. Have a great Brittany. All right, guys. Have a good one. Bye.